Hello, everybody. We had so much fun with synapses in class. We're going to do it again. Actually, this is one of those uber important areas of physiology to understand. So this video will actually only touch on one new area, and it's only 4 slides long, but just to go back and refresh you on the important aspects dealing with synapses. So I'll introduce to you a second kind of synapse, one we have not discussed yet. We'll look at the sequence of events again of what happens when action potential reaches the end of an axon, and then how is a neurotransmitter removed from the synapse and even why it's removed. So let's start there. Uh, the chemical synapse you've already seen, and we'll discuss that more in a second. I want to show you a different kind of synapse here at first, an electrical synapse. An electrical synapse, as you can see, looks fairly different than the chemical synapse in that you have these proteins called connexins that actually link the presynaptic and the postsynaptic membrane. In other words, if you imagine an impulse is coming this direction, and let me draw it on here for you. If you imagine the impulse is coming this direction, we need to pass the impulse from this side through these connexins to this side. The advantage of that, well, we'll look at some advantages in a second, but the fact that these things are really, really close together and a channel has been created for ions to pass through is going to give us certain advantages that we can touch on here. The two advantages are faster communication. Rather than having ions diffuse across a chemical synapse from the presynaptic to the postsynaptic side, have the ions travel from one side to the other, this can be a lot faster. Essentially, we're connecting the cytoplasms of two different cells. So if there's a action potential or a chemical change in charge on one side, it's very easy to travel this charge through to the other side and excite the other membrane and therefore carry out an action potential. It also allows for some synchronization. So it's going to be much easier for neurons to fire in a sequence if they're if they're joined very close together. So we see this kind of electrical synapse in some defensive reflexes. It's not something we're going to spend a lot of time on. It's far more important to really feel if you grasped the chemical synapse, but I wanted you to see the electrical just in case it came up. Okay, so let's go back and review the important parts, and let me move me out of the way here. The important parts of a chemical synapse. And all I want to do is just go back through these steps and show you again, just in case it wasn't clear in class, so that you feel like you've mastered this. Again, this was going to be a big issue in nursing because you're going to talk about different kinds of drugs and how they work and their mechanisms of action. And a lot of that concerns what happens right here in the chemical synapse. So again, I'm going to walk this through, and I have the major steps outlined, so just pay close attention. Here we go. So an action potential, an electrical charge is coming down from the cell body of a neuron. It's traveling to the very end of a neuron. It's traveling down the axon. When the action potential gets to the end of the axon, there are voltage gated channels. That is, there's proteins that are going to respond to the electrical change caused by this charge coming down the axon. And when the charge gets here, it's sort of like one of those electric doors. It's going to cause the proteins that are in the end of the axon to open, and in this case, allow calcium to move in through these channels. And I'll try to switch colors here periodically too. So the calcium is going to flow in, and that's sort of this step one. Now the axon is going to do something about that electrical charge difference, because now we have the synaptic vesicles. These vesicles contain neurotransmitter, and in this case we're going to call it acetylcholine. We'll talk about a different neurotransmitter here in a second. But because calcium has entered in, now these synaptic vesicles that contain neurotransmitter are going to move to the edge of the axon membrane, and they're going to exocytose their contents. They're going to spill neurotransmitter, again in this case acetylcholine, into the synapse. Synaptic cleft and synapse are the same thing. So now we have these little blue or greenish balls representing acetylcholine are going to move through the synapse, and they are going to bind to protein receptors on the other side. The term you'll see here is postsynaptic membrane. That could be a sarcolemma. That could be the membrane of another neuron on the other side. So here's the blown up version of it. The neurotransmitter, acetylcholine in this case, binds to one of these proteins, one of these receptors on the other side. And again, this view and this view are the same thing. When this neurotransmitter binds to the receptor, it's like a key going into a lock. Now, once again, we're going to open up the protein, it's going to slowly move open, and sodium ions, which are in the synapse anyway, are now going to flood through this sodium channel, and they're going to flood from here to there. Now, because they are ions, because they have a positive charge, when you get a bunch of positive charges moving from one place to another through a cell membrane, that's going to cause an electric change on the membrane as the charges move through. So now we have excited the sarcolemma. We've caused a change in the membrane on the other side. And for reasons we'll talk about probably a little bit later, because one region had this happen, the next region is also going to be more prone to open up, and so sodium will flow in and sodium will flow in, and we're going to get this charge is going to spread all across the sarcolemma very quickly, and this is the transfer of the action potential. The electric change, the electric charge from the axon has moved chemically by a neurotransmitter and excited the other side, and now we have sodium ions rushing through. We accomplish the goal if the charge changes on the sarcolemma. We excite the sarcolemma, and therefore we now have accomplished the mission. Now, we also have to clean up the mess too, because we actually have done more than just that. As the charge is dissipating or moving along here, and again, if this is a muscle cell, if this is a sarcolemma, then it's going to travel down those T tubules and cause the release of calcium inside the muscle fiber. But we have to clean up this neurotransmitter in the synapse. You see here, the neurotransmitter is degraded in the synapse. If we did not degrade the neurotransmitter, it's going to keep the doors open. Sodium keeps rushing in and keeps rushing in and keeps rushing in, and that muscle can't be ready to fire to the next electrical stimulation. So there's going to be an enzyme, and I'll type it for you here just to impress you. It is called acetylcholine esterase, E-S-T-E-R-A-S-E, and I'm sure we mentioned it in class. This is an enzyme that's going to erase or clean up the acetylcholine. It's going to break it down. And the components of the acetylcholine will either be recycled or they will diffuse out of the membrane. Potassium is also involved in this, and we probably won't delve into the details of that too much, but as the cell membrane is resetting on this side, potassium channels are going to open, and just imagine for a second that we have a potassium channel right here. Potassium is going to move out this direction, and that's going to help us even up the ion distribution a little bit. So just know, and we'll talk about potassium more with chemical synapse, with the transmission of a nerve impulse also, but just so you know that sodium is the main trigger here, but the potassium channels will also be on the membrane, and they're going to move out, and that's going to allow us to even everything up. Short way of saying this is, we can't create a mess without cleaning it up, and that's what we're going to do. We're going to recycle the neurotransmitter. We're going to put the ions back into place so we can then have another impulse come through. So if this is not clear, watch it again, because you need to be able to recreate these steps and talk about what's happening as part of your understanding of this topic. So let me squeegee everything here and go to our last slide. Which deals a little bit more with the neurotransmitter cleanup. And again, why? We'll go back to that point for a second. If there's not going to be a cleanup, then those doors are going to stay open, sodium keeps rushing in, and there can't be another electrical change in that membrane until we put it back the way it was. So how do we clean out the neurotransmitter? Some of it does just diffuse, it will just leave the synapse. Some of it's going to be broken down by acetylcholinesterase. The enzymatic degradation is just going to be the enzymes will break it down. And there's also going to be some reuptake by cells. And let me go back for a second to this. Again, here is going to be diffusion, which is, diffusion is going to be we're simply going to move out of the synapse like that. The enzymatic degradation is going to be where this neurotransmitter is broken down by enzymes. And then reuptake is where there's going to be channels on the axon side that are going to be taking up components of neurotransmitter and recycling them. I mention this because you will no doubt run across a discussion of SRIs, which are serotonin reuptake inhibitors. If we are talking about a different kind of neurotransmitter junction, well, a different neurotransmitter like serotonin, some people who are taking antidepressants like Prozac, they take that because the uptake is going to be slowed. Rather than us releasing serotonin and sucking it back up into the axon and stopping the reaction quickly, serotonin is going to be inhibited from going back up and it kind of lingers in the synapse a little bit longer. Serotonin is a happy neurotransmitter, so the longer serotonin is in that synapse, the more we're going to have prolonged positive sensations. So some people who are taking Prozac or other antidepressants are taking, they're called SRIs or SSRIs for selective serotonin reuptake inhibitors, but these are going to be ways that we can actually change or alter the chemistry of a synapse. So again, for this video, feel comfortable with how a synapse looks, what's the anatomy of a synapse, what's the physiology, how do impulses travel from one side to the other, and be able to contrast the two different kinds, electrical and chemical synapses. That will do it. We're done.